Hey everyone, and welcome to The Explainer. Today, we're diving straight into the fascinating college-level biological mechanics of how vaccines actually work. We're going to go way beyond the basics and look under the hood at the cellular and molecular levels to see exactly how these highly calibrated training simulators protect us. It's a pretty incredible biological marvel, so let's get into it.
Here's a quick look at our syllabus for today. We'll start with immune system basics, then move into how vaccines train immunity, look at traditional vaccine technologies, explore next-gen vaccine technologies, and finally wrap up with the power of herd immunity.
All right, let's kick things off with Section 1, Immune System Basics.
To understand the simulator, we really need to know the troops being trained, right? Human biology basically gives us two main defensive pillars. First up is the innate immune system. Think of these guys as your fast-acting first responders, your physical barriers, and your inflammatory responses. But the real star of today's explainer is the adaptive immune system. Now, this learning system is a bit slower to act at first, but it utilizes T cells for cell-mediated defense and B cells for humoral defense. And what's amazing is that this system allows us to learn, heavily target, and then remember specific threats for decades.
So what actually triggers this adaptive system? Well, it's the antigen. An antigen is essentially just a specific piece of a virus or bacteria, usually a protein or a polysaccharide, that our body recognizes as completely foreign. You can think of it as the ultimate biological red flag. It literally tells your immune system, hey, an invasion is happening right now.
And in response to those red flag antigens, our B cells, which are a type of white blood cell, kick into gear and start producing antibodies, primarily IgG. These are basically precision-guided proteins that are custom designed to lock onto and neutralize that one specific invader. And the best part? Once the infection is cleared, a few of these specialized cells stick around to keep watch. Boom! You are now immunized.
Moving right along to Section 2, how vaccines train immunity.
Okay, so the absolute crucial point you need to take away here is this. Vaccines are, for all intents and purposes, safe training simulators. They safely present that red flag antigen to your body without any of the actual dangers of a full-blown live infection. By basically running a highly realistic fire drill, they engage your adaptive immunity to build up a massive defense force. That way, if the real pathogen ever actually shows up, your body is already prepped and ready to fight it off.
Down at the cellular level, this whole thing triggers just a brilliantly orchestrated sequence. First, the vaccine is injected, usually right into your muscle tissue. Right there, specialized innate immune cells called dendritic cells scoop up the vaccine antigen. They act like little scouts, trafficking this antigen straight to your draining lymph nodes. Once they're inside the lymph nodes, they use MHC molecules to present little pieces of the antigen to activate your T cells. Those activated T cells then provide some absolutely crucial help to your B cells, telling them to differentiate into plasma cells that just pump out targeted antibodies, while also forming long-term memory cells that go and hide out in your bone marrow.
Now this perfectly illustrates why we need vaccines. Your primary immune response to a brand new antigen, like from a first vaccine dose, is actually pretty slow. It can take days or even weeks to achieve seroconversion, which is the point where protective antibody levels finally show up in your blood. But check out what happens next. Because of those memory B cells you just generated, your secondary response to the real pathogen later on is incredibly fast and just overwhelmingly strong. You neutralize the threat entirely before it can cause severe disease.
Okay, on to Section 3, traditional vaccine technologies.
Now that we know how the training works, let's look at how we've actually delivered these antigens over the years. For much of the 20th century, we basically relied on four main technologies. We used live-attenuated vaccines, which are weakened viruses, like the MMR vaccine. We used inactivated vaccines containing completely dead pathogens, like the polio shot. We used protein subunit vaccines that only contain very specific protein pieces of the virus, like hepatitis B. And finally, toxoid vaccines, which use inactivated bacterial toxins. Think of your tetanus shot for that one.
When we compare these methods directly, we see a really fascinating biological trade-off. Live-attenuated vaccines trigger a massive, enduring immune response, honestly, often providing lifelong immunity with just a dose or two. And that's because they actually replicate slightly in the body, which perfectly mimics a natural infection. But because they're alive, they carry a very rare risk of reverting to a virulent form, making them unsafe for folks with weakened immune systems. On the flip side, non-live or inactivated vaccines are incredibly safe since they absolutely cannot replicate or revert. But the trade-off? Their weaker immune response means you're going to need booster shots to keep your memory cells sharp.
And here's an interesting problem. Because those non-live and subunit vaccines don't replicate, they sometimes really struggle to get the immune system's attention. It's almost too quiet. To fix this, scientists add something called an adjuvant. An adjuvant, which might be something like aluminum salts or newer compounds like AS01, basically acts as a loud, danger signal to supercharge the innate immune system. It ensures that those scout dendritic cells get properly activated, which then leads to a much more robust adaptive immune response.
Next up, Section 4, Next-Gen Vaccine Technologies.
Fast forward to today, and we are engineering completely new ways to deliver immune training. First up are viral vector vaccines, which you probably know from the Ebola and COVID-19 vaccines. Here, scientists take a completely harmless carrier virus, like an adenovirus, and essentially use it as a Trojan horse. They alter its genome to carry the genetic code of the target pathogen's antigen straight into our cells. Because it mimics a real viral entry, it triggers amazingly strong T-cell and antibody responses without even needing an adjuvant. The only catch here? Well, if your body already has immunity to the harmless carrier virus, your immune system might just destroy the Trojan horse before the training can even begin.
But we can go even more advanced with mRNA vaccines. Here, we completely bypass using a virus at all. Instead, we take fragile genetic constructions, messenger RNA, and encapsulate them inside microscopic lipid nanoparticles. These little fat bubbles seamlessly fuse with our cells, dropping off the mRNA. Then our own cellular factories, the ribosomes, actually read this code and manufacture the target antigen protein ourselves. Once the protein is made, the immune system spots it, sounds the alarm, and builds the defense. The mRNA safely degrades right after. And no, since there's no reverse transcriptase, it literally cannot integrate into your DNA. Plus, because we don't have to grow live viruses in massive vats, these are incredibly fast to manufacture.
Looking at this data, we can see exactly why having a diversity of platforms is absolutely crucial. Different bugs require different weapons. For example, if we want to defeat highly mutable viruses or intracellular pathogens, we really need the strong cellular T-cell responses that are generated natively by mRNA or vector vaccines. But if we're fighting a toxin-mediated disease like tetanus, that can be entirely handled by a simple toxoid vaccine driving a strong antibody response.
Finally, let's zoom out for Section 5, the power of herd immunity.
Okay, we have to address a really common and honestly incredibly important question that causes a lot of confusion. Can a vaccinated person still catch the disease they were immunized against?
The short answer is yes, but there is a massive biological distinction we need to clear up here. Most vaccines are just incredibly effective at what we call disease prevention. This means your vaccine-induced memory cells neutralize the pathogen before you experience severe symptoms, hospitalization, or death, even if a mild infection briefly takes hold in your body. However, the true holy grail of vaccinology is sterilizing immunity. That's when the immune response blocks the infection so completely and so rapidly that you literally cannot acquire it at all, and therefore there's no way you can transmit the pathogen to anyone else.
And that specific ability to halt transmission leads us straight to the concept of herd immunity. For highly contagious diseases like measles, we rely heavily on herd immunity to protect infants who are just too young to be vaccinated and folks with compromised immune systems. But because measles spreads like wildfire, a staggering 95% of the entire population needs to be vaccinated just to break those chains of transmission and keep the vulnerable safe.
When communities actually step up and hit these critical public health thresholds, the global impact is just staggering. To put that into perspective, the World Health Organization estimates that current immunization programs save roughly 2 to 3 million lives globally every single year. Let that sink in. It is arguably one of the most successful public health interventions in all of human history.
As we've seen throughout this explainer, whether we're talking about a 19th century weakened virus or a cutting-edge 21st century lipid nanoparticle, the core principle is exactly the same. All vaccines fundamentally rely on the absolute genius of our own biology, our highly evolved adaptive immune system's incredible ability to learn, remember, and protect us. Which leaves us with a pretty thrilling question to ponder. As our understanding of immunology, atomic-level structural biology, and mRNA engineering continues to accelerate at just a breakneck pace, what devastating disease will we relegate to the history books next?